today. Concerns, topics you want to have more information on. Today we're starting genomes. We've gotten past chromosome structure, and we're going to move on a little bit into what's bigger than a chromosome. That's the set of all of the DNA that's in your nucleus, your genome. Bless you. You're all too kind. Okay, so when at some point have you heard the word genome or genomics mentioned? In what context? What's it about? Why is it interesting? Why do people talk about it? Do you care about your genome? What's your genome about? So it's all of your chromosomes. Why is it interesting to study the whole genome, all the genes in the genome, all of the nucleotides that make up your chromosomes? Has anybody heard of 23andMe, the company? Ancestry testing. You do a cheek swab or you spit in a tube until you fill up the tube, which takes an amazingly long amount of time to fill up a tiny tube with saliva, it turns out. We should have a race someday in class, maybe. But you send off your DNA, and the company basically sequences a lot of the DNA in your genome, and then they can make predictions about potential health issues you might have someday, and definitely something about your ancestry. That's one of the reasons that a lot of people are interested in the genome as a whole. It's all of our DNA. Some of the DNA makes genes that encode proteins that do the job of keeping us alive. And other parts of our DNA do other things, like telomeres, the ends of the chromosomes, which is DNA sequence, basically keeps our chromosomes from unraveling. And there's some discussion, although I think it's been pretty much debunked at this point. There was a thought in time that as your telomeres shortened, cell division after cell division, that maybe that was what caused aging, was your chromosomes started to get eaten from both ends. They degrade as you age, and that was part of the aging process. And it's kind of still unclear whether that's true. That's part of your genome as well.
Is it true that some animals or some organisms might have longer telomeres?
Well, so telomere length, which we'll talk about eventually, not necessarily in this chapter, does vary, and actually you start out with long telomeres, and your telomeres get shorter and shorter as you age, which is curious. All right, so let's see how we're doing with today's entry quiz. Again, structure of the class, same as always. Hopefully you tried to access some of the material beforehand, the readings in the OpenStack textbooks, and watch some of the videos. We're going to find out whether or not that's true, generally speaking. So, plants, amphibians, or bacteria have a larger genome than humans. Select all that apply, which I don't think some of you caught on to, but that's okay. Mixed results. Plants have, now how many plant species are there in the world? 300,000? I don't know. It's a lot. Definitely a lot of species. Okay, so this is not all plants. Are there plants that have a larger genome than humans, really? Absolutely. Lots of plants have way bigger, that is, they have more nucleotides in their genomes. If you tally up all of the nucleotides, all the base pairs on all of the chromosomes in a plant nucleus, a lot of plants have more DNA than we do. Same is true for amphibians. Some have smaller genomes than we do. Some have larger genomes than we do. But there are some that have larger genomes than humans. More DNA that encodes various things like proteins. Are there any bacteria that have bigger genomes than humans? Not that I know of. Bacterial genomes are really small, and we'll talk about that as we move through the semester. So the correct answer here would be A and B. This is related to one of the videos that was for class today called the C-value paradox. Can anybody briefly summarize, let's take a show of hands, who wants to offer, what is the C-value paradox? In genetics, there's something weird about genome sizes and the organisms they're in, which is sort of outlined here, especially in A and B, for those of us humans that think we're the top of the animal kingdom. What's the C-value paradox? Sir?
It's
Okay. So I will take that. Thank you very much. So yeah, the idea here, go ahead if you want to add.
Is it because the more complex an organism is, the more genomes it will have?
Yes, both are accurate. So let me summarize then, if you didn't, especially if you didn't hear, and so that it's clear on the recording. There's this anticipation decades ago when people were just starting to be able to sequence genomes, like starting to get an idea of what the size was in terms of millions of base pairs, bacterial genomes, amphibian genomes, plant genomes, and mammal genomes were. And this wasn't by sequencing. The earliest experiments were done by actually extracting DNA from cells and just weighing. Like what's the mass of DNA that you get out of a certain number of cells? That's how they figured out what the relative sizes of the genomes were, decades ago. And decades ago, humans thought we were, top shelf, we're the pinnacle of evolution, we're the most highly evolved, and then that comes to this concept of most complex organism. So humans thought, well, we're really complex because we can talk, we can communicate verbally, we have written language, which no other species presumably has. We can anticipate the future, we can plan for the future, so we're the shit, in the good sense. But then when people started measuring genome sizes, they were taken aback a little bit because, wait a second, if we're so awesome, we humans, then how is it that there's some plant, they can't even move, yet they have more DNA than us? So this was the paradox related to complexity, our perceived complexity, which is biased. I mean, who's to say what organism is more or less complex? But there was an anticipation by scientists, and there may still be, that complexity should scale with genome size. So if you had a definition of complexity means, you might say on the x-axis have genome size, right, independent variable, how many millions of base pairs are in the genome. Sorry about that. Back to this. So x-axis, you might have genome size, y-axis, you might have some measure of complexity. And if it's true that the number of nucleotides in your genome is related to your complexity, then you should see some sort of a relationship like that, where the bigger your DNA, the more DNA you have, the more complex an organism you are. But that doesn't hold up. And that's the C-value paradox. Why do we have so little DNA compared to an amphibian? Any questions about that? There was an expectation. It's not a reasonable expectation, I don't think. And we'll talk a little bit about why there is no correlation between complexity and genome size as we go through the semester. And the other question was, boring question, but we're moving into computational sequence analysis, is why everybody has a tablet, smartphone, laptop. We're going to do some of this today. Yes, FASTA formatted DNA sequences starts with that symbol. Whether that's greater than or less than, I can never remember. It's one of the two of those. It's not an @ sign, it's not a pound sign, or to you or me, a hashtag, and not a period. It's option C. So good. So today, we've got these topics to discuss and into next class as well. One of the main tasks, again, with you having computers is, if we're going to be information literate about genetics, if we're going to know something about how to look up genetic information, let's start with something simple. Look up the DNA sequence of a gene, which was a task that we had for today in one of the exercise that I asked you to try for today. And then also something about comparing genes between different organisms, either between two different species or between two individuals of the same species, like you and me. And that's this DNA alignment consensus sequence task that we're going to work on a little bit as well. And ultimately, by the end, hopefully we figure out, if we compare any two of our DNA sequences, on average, how similar are we in terms of DNA sequence? So that's where we're heading. One other point. Although you do not have to do this, I just wanted to make it known as a resource. I just made a new video on the genetics of ancestry testing. So if you are interested in how genome sequencing and your genetic information is used to try to figure out where you're from in terms of your ancestors, then this is one of certainly many videos that you could use if you want. Not an assignment, just a resource. Okay. Something's funny here. So one of the papers that was in the chapter was this King and Wilson 1975 paper comparing humans and chimpanzees, our closest relatives. So we can have a separate discussion later if you want about evolution, but chimpanzees and humans are sister species. They are the most closely related species to us on the planet, but this was the paper so many decades ago that proved that point. And it was all about comparing genomes. This is one of the earliest comparisons of sequence information from two different organisms. So we've got the chimpanzee on the left, and we've got, who's on the right? Shown there with an X-ray crystallography experiment of the structure of DNA. Our example of Homo sapiens is Rosalind Franklin, who was, along with Watson and Crick, not so credited for all the work that she did on this, because Watson and Crick got most of the kudos for doing this, but she was also highly involved in that. So humans and chimpanzees, are we a lot more complex than they are? I don't know. She was pretty smart, but so are chimpanzees. So data from their paper, 1975, the title of the paper, Evolution at Two Levels in Humans and Chimpanzees. So what did they do? They took, not DNA in this case, so I should have maybe shown you this after we talked about translation and started talking about proteins or polypeptides, but they took the same proteins from humans and from chimpanzees. These are the proteins listed here. What's your favorite protein on the list? What's a protein you recognize?
Hemoglobin.
Okay, they studied a lot of hemoglobins. So what's hemoglobin? The protein that binds oxygen in your blood cells. Okay, so that was a highly abundant protein that they could easily get from chimpanzees. Blood draw, chimpanzee, take blood from a human, extract this specific protein or these individual members of this class of proteins. And then they just figured out what were the amino acids that made up each of the chimp version of hemoglobin and the human version of hemoglobin. What did they find?
Very few differences.
Very few, as in, for hemoglobins, the differences between human and chimpanzee was essentially zero, except for one of them down there. There was one amino acid difference out of how long was that amino acid, or how long was that protein, number of amino acids. Hemoglobin delta, they saw one amino acid difference between humans and chimps, and it's 146 amino acids long. So that's less than 1% difference. Same protein, two different organisms. Chimps, their protein is basically identical to ours, except there's one amino acid difference. Right? So this was the first time, and that's a pattern across most of the proteins that they studied, was that when you compared us and monkeys, we're pretty much identical at the protein sequence level. And then later, as DNA sequencing technologies became more easy to use, more affordable, then people started looking at DNA sequences and found the same thing. If you compare the DNA of humans and any organism on the planet, humans and chimpanzees are the most closely related. 99+%. It's the same. It means if you look at our DNA sequence of any part on any chromosome, out of 100 base pairs, chimpanzees have the same sequence with one change, one G, T, C, or A different. Any questions or concerns about that? So we're fancy monkeys, basically. We've evolved different things since humans and chimpanzees had a common ancestor, like the ability to talk and the ability to have written language and other genes that are important in several aspects of our biology. But for the most part, we're pretty similar. We had this sequence alignment exercise, and I'm wondering if anybody has any questions on that. I'm going to switch over to the laptop so I can actually show you what we were doing. Hopefully you all tried this, and if you brought any questions, that's fine. We can go through it now. We can solve the problem and get the answer, or if nobody has any questions, we can skip right past it. So it looked something like this. So in this case, you might consider that these are 5 different sequences from a similar part of the genome of one organism. Let's say it's humans. So did anybody solve this problem? Okay, so let's see. How do you go about solving this? So we need to take these sequences, they're all related to each other somehow. We're going to see how they are if you haven't already done this. These 5 sequences are all from the same gene, say. So we just need to move these, and this is why it's useful to do this in a spreadsheet. Slide these rows from sequence 1, sequence 2, until they all line up. So for those of you that succeeded, how did you approach solving this problem? What do you look for? Yeah, go ahead.
I just wrote on another piece of paper and
Yep.
try to find some similarities.
Fantastic. So the first thing to do is to look at all these 4 sequences and try to find something that's similar, a similar sequence in all 5. So is there a small, we call that a motif, M-O-T-I-F, a DNA sequence motif, a short little stretch of nucleotides that are all the same that tells us something about how related these are. So in here, is there one set of nucleotides that's common to all 5 of those sequences? CTT. Okay, so let's say we want to align all these sequences on CTT. What do we have to do to that row, the top row, to get the CTTs to line up? We need to go right a little bit, 2 cells. Is that far enough? Nope, one more. Okay, so now hopefully you see that there's a common sequence in those 2 sequences. ACTGCTTG. It just wasn't obvious because the sequences weren't lined up left and right next to each other. So these 5 sequences are all from the same spot in the same genome of an organism. And this is how scientists did those initial experiments, both with amino acid sequences but also with DNA sequences. They got random DNA sequences. That is, the DNA sequences don't all start at the same spot on the chromosome. They have staggered start and staggered ends. Our job, or a computer's job when we do this in the laboratory, is to move the sequences left and right relative to each other to try to find a similar sequence, a motif. So what do we have to do with that batch to line it up with the one below, sequence 3? CT, yeah, we'd have to move it left, but that's not easy on a spreadsheet, so let's move this one right. So now I've lined up the CTT again. Does anybody have any concerns about what's going on? There should be a lot of concerns. There's some weird stuff going on. So, sir?
Can't even see the assignment.
Where did you?
Google Classroom.
This was from the course manual. It was the assignment I assigned for doing today, but it was one of the fail assignments from last class. There's a link from the course manual to do this. Yeah, so if you go into the course manual, click on the link, you'll be asked to make a copy of this assignment. So I'll give everybody a couple minutes to work on that. Questions? Yeah.
I moved every other sequence in respect to the first sequence. So how's that gonna, is that gonna create a difference in my consensus?
Not necessarily, no. So as long as you wind up with the same relative positions of all of these sequences, that's fine. And because all of these sequences are random, in the sense of where they start and where they stop relative to each other, it's fine that there are some parts that are hanging out by themselves. If this DNA sequence had been a little bit longer, if we had actually been extracting DNA from an organism and sequencing it, if we had gotten the next 4 nucleotides of sequence, it would probably be A, G, C, C. Really important to do the exercises before class. Other questions or concerns? Is anybody having technical issues? Because I'm happy to come briefly do some troubleshooting.
Can there be many motives?
Great question. So
I
there,
got TGC as one.
yep. So let's see, if we move this one, there's TGC, which becomes a motif. That is, it's shared among most of the sequences, not necessarily this third line. But there's also, where is there also TGC? There's not just this one,
There's also a second one.
there's one there too. And that's one of the reasons that it's worth doing this sort of an exercise to try to catch some of these issues that you might run into when you do these sorts of comparisons of DNA between different organisms, different individuals or different species, is that you could accidentally, or you could purposefully, line them up on that TGC. So if I finish, just for the sake of getting this alignment the way I like it, this is, in short answer to your question or to your point, this is another one of those cases where there could be multiple solutions to this problem. This is a puzzle. It's like a crossword puzzle. So the best that geneticists can do is try to program computers to, with all the rules that we would follow to do this sort of a thing. So in this case, I found a solution where at every nucleotide position, all of the sequences have the same letter. T, G, C, T, T, G, C, G, A, and so forth. So if I were trying to figure out whether or not humans, chimpanzees, how different humans, chimpanzees, gorillas, orangutans, and macaques were, if I got DNA sequences from the 5, I'd want to find some motif, some sequence that's common to them, and then look for those rare differences between the genome sequences. But what would happen if I took that CGC, let's color it yellow just to make it really clear. Wait, was that the motif you were saying was, which one was the one we had two of? TGC. There it is, TGC, thanks. So if we color that yellow to make it obvious, what would happen if we lined it up? What would happen to the beautiful identity, the similarity of all these nucleotide sequences if we accidentally aligned that or purposefully aligned it with the other TGCs?
It would make smaller.
So there are two TGCs here. So if we accidentally or purposefully moved that TGC, so now it lines up with the other members of that same sequence. What happens down here? Is there similarity in every column? Do they agree? All the sequences have a G, all the sequences have an A. No. This is actually less perfectly aligned. Not all of the nucleotides in these sequences agree with each other, which suggests that that top sequence is misaligned. So the goal is slide the sequences left and right until you find the best alignment. This is called a multiple sequence alignment. Slide them all left and right until you find the best match of all the sequences you're working with. And that's when you've reached the conclusion. And then you can compare different sequences to each other. And we'll see in some cases it's absolutely possible for there to be a mismatch. So if I go back to this, what would it mean if I, instead of here, sequence 3, if instead of a T there was an A there, what would that mean? 4 out of the 5 sequences have the same nucleotide there, a T. This one has an A. Why could that, how could that be? SNPs, mutations. So it might be that in this species, whatever species it is, species 3, that it does differ from all the other species because there was a random mutation that makes that species distinct from all the other species. And it's these sorts of polymorphisms, which just is a fancy word for changes, differences, that are also used in ancestry testing. So there are some mutations, differences, that distinguish different populations of humans. And geneticists know which of those differences are indicative of where you came from in the world. So when the company receives your DNA and tests, they actually don't sequence all of your DNA. They just look at the very specific letters in your DNA that they know are distinctive of where you're from. Whether it's Inuit or Polynesian or Han Chinese, African, Norwegian. That's why this is important to be able to do, to take different sequences from different organisms and align them, then look for differences. It gives us something, tells us something about mutation rates or the genetic differences between species like humans and chimpanzees. All right. So human-human differences. So far that's just human-chimpanzee differences and a little bit of a look at how DNA sequence alignments work. So this was another paper that was part of the assignment for today, just taking a look at, this is from the course manual, Levy et al. The first time that an individual human's genome was sequenced from start to finish. When was this? 2007. So it's been a decade, a little bit more. So how large is the human genome? Did anybody figure that out? You Google how large is the human genome. It should give you a pretty general answer. Should tell you that it's about 3 billion nucleotides, or 3 billion base pairs. We got 3 billion nucleotides in our genomes, and the point of this paper then was they wanted to know what does one person's genome sequence look like and how similar are different humans to each other. So this is a good point to do a little bit of review. This was published in that paper. Hopefully you downloaded it, the PDF, and looked at these figures. So this is what sort of a figure? Vocabulary. It's a karyogram. Yep. It's the photograph of the chromosomes, not the karyotype, which would be the numerical 46XY would be the karyotype. Oh crap, I gave away the answer. Was the donor DNA male or female?
Male.
Male, because there's a Y chromosome down there. Okay, so this was the genome sequence of a single individual, a human male. And how is this genome sequence different from those that had been used previously? The first human genome sequence was published in 2001 or 2002. So this was about 5 years after that. People already had human DNA sequences. But the first human genome sequence that was published was a random bunch of human genome sequences. They had taken DNA, in other words, from a bunch of different humans that were all anonymous. They were all anonymous DNA donors. They sequenced all their DNA, and they did kind of like that sequence alignment thing, where they sequenced all the DNA, all the chromosome sequences, they lined them up to each other, and then they created what is a consensus sequence. So you can imagine that DNA sequence alignment thing we just did. They might look at the same sequence from 100 different donors, and they might get something that looks like this. I'm just going to draw 3, just for example. So let's say this is the DNA sequence in the same region of the same chromosome for 3 different people. The goal of scientists was to make the reference human genome sequence so that we could all study our genes and say, what version of the gene do we have? What is the human genome sequence? Is there the human genome sequence? No, because we're all unique. So every individual person has their own specific DNA sequence. Even individual twins are slightly genetically different. So this first approach, they basically just needed a lot of DNA to sequence. So they got DNA from a bunch of different people. So in the reference human genome sequence, which is what was initially published, what is the letter here at this first position? What do you think they said was the normal nucleotide at that position? An A. They said, we're going to take the average of everybody's DNA sequences, and we're going to say that is the nucleotide that's They didn't say this, but they imply that's the DNA that's supposed to be there. That's what most people have, is an A at that position on that chromosome. So you just take the average, and this is what's called a consensus sequence, because it's the consensus, it's an average. And that's why these multiple sequence alignments are important, because that's how they lined up the DNA so they knew that they were looking at the same parts of the same chromosomes, and then they can create the average consensus sequence. So that was what was published in 2001, 2002. The first human genome sequences was an average. So this was the first time that a single individual's DNA had been sequenced. All this DNA came from this one guy, J. Craig Venter, who basically did this as a publicity stunt. He was leading, so at the time, this is snooze for 3 minutes if you want, because I'm going to talk about history for a second. Just for context. If you don't care, that's fine. Craig Venter was competing against the US government to sequence the first human genome in the early 2000s. So it was the National Institutes of Health, the National Human Genome Initiative, was trying to do a public version of this. And then Craig Venter got a bunch of people to give him money to try to do a private version of a corporate, let's sequence a human genome and see if we can beat the federal government and all their resources at it. So he was the head of that company, and later then he decided, as probably another publicity stunt, I'll be the first individual human whose genome is published. And so that's why he is the guy whose genome was the first complete individual human genome sequenced. Why would that be, well, we'll talk about this a lot during the class, but why would that be good or bad? Would you want to have your genome sequence published in a scientific journal and put in a database online? I see a couple heads shake. Some of you are thinking about it. What's one pro and one con? What's one good thing maybe about sharing your DNA sequence with everybody, and maybe one bad thing about doing it? Could I do something nefarious with your DNA sequence, in other words?
Well, one pro is because he was hashtag first. He was like, I was the first one to comment on this Instagram photo, but with my genome sequence.
Right. Okay, so there's notoriety or publicity. There's some value with sharing DNA sequence because the more sequence that doctors have available, maybe, for example, the more they can learn about different diseases. So if they have a bunch of people who don't have a disease and we have their genome sequences, and we have another category of people that have shared their genome sequences with us and they all share the same disease, then we could do something like that sequence alignment. We could take all the wild type people and all the disease people and align all their DNA and see if there's something that's common to everybody who's got this disease that nobody that's normal, undiseased, has. And then you could try to figure out the genetic basis of human diseases that way. What's a reason not to share your DNA sequence?
Use it on a voodoo doll.
Pardon?
Use it in a voodoo doll.
Yeah, sure. Cloning, somebody gets your human no, they can't do that yet. It's often.
Isn't that something about it's one of the issues that some people have with 23andMe? Once you give up your DNA, you don't have the rights to your own DNA anymore?
So that's an interesting point that we should definitely discuss more, is who owns your DNA sequence? Can DNA sequences be copyrighted or trademarked? Because they're naturally occurring, and there have been some famous cases, including most famously a gene we're going to talk about today or next time if we get to it, the breast cancer 1 gene. The gene that, if you have a mutation, makes you susceptible to breast cancer. The scientist that first sequenced that gene and discovered that it was predictive of breast cancer risk was an academic scientist at University of Washington, then at Stanford, I think. Doesn't matter. A company then came along and said, we want to patent a DNA test that your doctor will give you to extract your DNA and test for this one mutation or a series of mutations that would predict your risk of getting breast cancer. So the academic professor said, it's a DNA sequence, everybody's got it, you can't patent it, it should be free for everybody. But the company said, well, we developed the specific technology to do the reading of that gene, therefore, so it has been settled in court now, but there was a huge issue at the time of, it's a naturally occurring thing. Genes, they're in all of our cells. Nobody created them. Well, we could discuss that as well. No human created them. So, yeah, that's a big issue. The other thing that people raise is,
There was that issue with the HeLa cells like 60 years ago. She didn't give consent to give her like prolific
Right.
cancer cells, and like people are making money off of testing on them, and her family has no rights to any of the money that they've made off of it.
so there have been a number of cases in science like the HeLa cell line, which is cell biology, not genetics, but it's the same point, is that if you give up your rights to something, if you make something publicly available, then if somebody makes money on it in the future, do you get a cut?
No.
Probably not. The biggest issue that people have mentioned with getting DNA testing and making it available is prediction of disease risk. So if an insurance company, which is they're always the bad guys that come up in this hypothetical story, if an insurance company gets your DNA sequence and they can read your DNA and say, well, you've got the SNP that makes you predisposed for breast cancer, then are they going to insure you?
No.
Or are they going to make you pay more? Or if they can see that you've got a genetic predisposition to Alzheimer's disease or narcolepsy. I mean, there's so many things that are genetically controlled. You can tell, not 100%, but you can predict whether or not somebody is at risk. Right? That's different than saying you are or you are not. A lot of these DNA sequences in our genes can predict whether you're likely to get a particular disease. But even that, and we'll see an example of that in just a little bit, even that can be too much. So a lot of people are very careful about how they share their DNA sequences. So, good timing. Skepticism, information literacy, and evidence-based decision-making. My passion, really, in teaching genetics and teaching biology at all, teaching science at all, is to help generate people that are willing to make decisions based on fact and evidence, and not just randomly pick what stock they're going to invest in or what they're going to eat for dinner if they don't know if it's good or bad for them. You try to collect some data and then make a decision based on the data you have. What should you do? Not every decision like eating out necessarily. And one of the most critical strengths I think a lot of practicing scientists have is their ability to be skeptical, or maybe their willingness to be skeptical. So presumably at some point most of you have been told don't believe everything you hear, or don't believe everything you read, or hopefully certainly not don't believe everything you've seen on the internet. Fake news. And by the way, this is not going to be political. I just wanted to use that because that's a prime example of why we need people that can make evidence-based decisions and know whether you're looking at something that somebody made up or if you're looking at something that's factual. So part of the course is going to be sort of highlighting that aspect of being a scientist. It's genetics framed, but these are the core values or core objectives. So for example, this is one of my favorite websites, Spurious Correlations from tylervegan.com. This is a fantastic website. If you're interested in this, go, there's a ton of these. So what are we looking at here? It's a graph across time of two different things. What are we looking at?
The birth rate in Maine and the per capita consumption of margarine.
Yes, so what do you see, the blue or whatever lighter color that is the divorce rate in Maine. And right on top of it, in whatever the other color is per capita consumption of margarine in the US.
I can't believe
What
margarine.
does that mean? What does this mean? What does this tell us if we're going to make decisions based on information?
Margarine causes divorces.
Margarine causes divorce. That's what it's, so is it true? Let's take a show of hands. Is this true? Does consumption of margarine cause an increase in divorce rate? Raise your hand, yes? No? Why not? The data says it's true, right? What's wrong with my claim? It's extremely broad, it's what? Heard something about published?
Correlation doesn't equal causation.
Correlation is not the same as causation. Because two things are similar doesn't mean that there's a relationship between them. That's exactly what spurious correlations is all about. He just mined all these publicly accessible data sets and looked for the strongest overlaps of random data. If you had to make up a hypothesis, what would your hypothesis be if there really was a correlation between consumption of margarine and divorce rate in Maine. This is important to practice. It's silly, but coming up with hypotheses is a really important task, and this is just a way to practice.
Margarine's a comfort food,
Margarine's a comfort food, so
or
you...
divorces, you eat more butter.
So it's the opposite correlation. When you get divorced, then you wind up gorging on butter, and you've got sticks of margarine in the fridge, and you're just chowing down. Well, but maybe. So it's a hypothesis. Now then I would ask you, though, why just in Maine? Why is this only true in Maine? Shouldn't we be looking at the per capita consumption of margarine in Maine
Yeah.
if it's correlating with divorce rate in Maine? Right? These are sort of details that whenever you're faced, I'm just imploring you. I'm just trying to show you some examples. If you see something like this, be skeptical. Like, what's on the axes? Why is it Maine versus US? And most importantly, if you take any note about this, most importantly for this class and for the rest of your progress as a scientist, it is, does the hypothesis make sense? Does it, intellectually to you, without any other information, does it make sense? So this example, another one from the same website, total revenue generated by arcades on the y-axis here, dollars in millions, versus the number of computer science doctorates awarded in the US. Really high correlation. Right? I've got an easy hypothesis for this one. Computer science doctorates go out and make more video games that people want to play. So the more PhDs in computer science you put out, the more video games there are to play, more money the video game companies make. I don't know, that's a hypothesis. It makes sense to me. So if somebody told me that this is causative, a causative relationship and not just a correlation, maybe I would think that was reasonable. Maybe not. Your turn. This is a paper published in Association for Psychological Science, the Psychological Science Journal, 2013.
My hypothesis for this would be that subconsciously red is a sexual color, and at peak fertility, you want to get your freak on.
Yep.
So women will
Sure.
wear it at peak fertility.
Okay. Other hypotheses? Do you think this is true or not?
I don't think it's true.
Anybody? So I've got one, don't think it's true. For those of you that are women, and I hate to put you on the spot, but this is about women, do you think this is true? Do you do this, if you're willing to admit it?
I wear red every day because I go to Fresno State.
So this is, yeah, it's supposed to be humorous, right? This is an interesting example. It's actually very serious, but at the same time it's humorous. So we've got an alternate hypothesis up here. What I didn't tell you is that this study was based on a survey that these psychologists went to, I think it was a university in England somewhere. And they just stopped people on the street, like they do over there by the library. They try to hand you stuff. They stopped a bunch of people on campus and asked them to fill out this survey about when was your last menstrual period and what were you wearing and all this stuff. And that's how they got the data set for this study. And then a whole bunch of people erupted in laughter at this study and said, this has nothing to do with causation. This could be just all correlation. Because, for example, if you're at Fresno State and somebody came and did this study, what's the probability that you're a woman and that you are at that time of the month and you happen to be wearing red because that's one of the colors of Fresno State? Right? It might have nothing to do with fertility. It might just be correlation that you're always wearing red because it's your favorite color. And then sometimes it's correlated and sometimes it isn't, but that looks like a causative relationship. Right? So even when you see something in a journal, you still definitely have full license to be skeptical and ask, does this make sense? Because even if it's published and you think, oh, it's published, it must be fact, if the hypothesis doesn't intuitively make sense, then you're certainly allowed to be skeptical and say, that can't possibly be true. But instead of just saying that, you have to design an experiment and test your hypothesis. to close. We've got a new assignment. We're going to do this through the semesters for small numbers of points, but I'd love your participation. So this is important for building some of your scientific literacy and information literacy skills. The last time I was at the grocery store, I was thinking about this, and I saw the newsstand, and I was thinking about being skeptical, and so my eye landed on which one of these?
Your gene.
My Genes, 100 Things You Never Knew from National Geographic, a pretty reputable, not a scientific journal, but a pretty reputable source of news. If you don't know about National Geographic, they publish a bunch of stuff about culture and science around the world, and they're pretty cool. And they've got a lot of good genetics tools on their website also. But I thought, okay, first of all, if I'm going to teach genetics, I probably ought to know what the 100 things I ought to know are. So I bought the $17.49 100 things I was supposed to know. Yeah, I did.
That's expensive.
It is expensive for things you should already know. Or at least things we're going to know by the end of the semester. So I think what would be really fun, but this is just me, so I'm asking you to participate for small numbers of points. It would be fun to fact check. Is any of this fake news? So I read through the 100 things, and I'm going to tell you just a couple things about them, and then we'll end. Just to reinforce this, though, the reason that I want to do this is because the student learning outcomes on the syllabus say that we're supposed to be working on efficiently finding and using quality information relevant to genetics, that is, fact-checking, looking at primary literature sources and knowing, is what somebody's telling me about genetics true, or did they just make it up? Can we find a source that says this really is a fact? And then also representing arguments opposing both pro and con different aspects of genetics related to society, which a lot of these 100 things have to do with. The thing that really pissed me off about this was that even though this whole thing, it's about that thick, it's about 1/3 of an inch thick, is just about the 100 things you never knew. It's not a normal National Geographic with articles about other stuff. So they definitely had space if they wanted to for the author to cite all of the resources they used when they came up with these 100 facts, but they didn't. So this is just facts. We're supposed to take it for granted that this is how DNA works. And they're giving this to whoever wants to pay that much money for it at the newsstand. So let's help the public figure out how much of this is bullshit and how much of this is true. So over the weekend, because I'm a nerd like that, I take a look at all the 100 facts, and I discovered that 81 of them specifically align with things we're learning about, topics we're going to learn about this semester. Gene regulation, transcription, translation, cloning. So of those 81, some of them are just easily, we don't even have to do any work to figure out if it's true or not. The human genome is made up of more than 3 billion base pairs. That's too easy to bother with. Some of the things that they said were facts aren't really facts, or they're kind of weird. Like, people say that Rosalind Franklin's X-ray crystallograph was the best one ever taken of DNA. I don't know if you can find a citation for that. Somebody probably did say that, but I don't know how we would figure that out. Or there are 10,000 different genetic conditions that cause serious health problems for those who inherit them. Sounds like a fact. But is it exactly 10,000 or approximately 10,000? And how would we figure that out? So some of these, in other words, I'm telling you, it's probably not the time trying to figure out if they're true. So only 39 of the 100 will we see this semester. We've looked at one today. So we humans share 99% of our genes with chimpanzees and bonobos, another organism. So what I'm asking you to do for this one, this is an assignment, so it's worth points, just 1 point, is right now on Canvas there's a discussion board that just opened that's called, down here at the bottom, 1 human chimp. That's because this is the first fact in the 100. The human-chimp genetic relationship. So just write me, please, a brief, 1 paragraph or less, just a paragraph, whether or not you think this is true or false, and support your opinion, fact, true and false, with 1 scientific publication that supports your point. Okay. These are going to be due not just before class, but by tomorrow night at midnight, so Thursday night, midnight, as Friday is turning into Friday. Just because I'd like to have time Friday morning to review your submissions so I can give you feedback in class on Friday. We won't do this every class, but there are some of these pseudo facts or maybe facts that would be worth checking out during the semester. Okay? So we'll get to the exercise next class. And there are 2 more exercises. Course manual, 3.2 and 3.3. Try those for next class as well. Try them.